is actually interfering with the natural growth of the food cycle, and that is affecting nutrition and food habits, because sometimes we are made to eat those food which are not natural to me. I should be eating food that is friendlier to my environment, which is produced in my environment. For me, a sour orange may be good than a very ripe mango. But anyway, there was a challenge for the policymakers because millions and millions of people are starving. There is hunger. So it is our duty to actually create food, have surplus production, create nutrition. In that process, we generally intervene with the nature by food engineering, by modern development, by agricultural technology, by so many things. And on the one side, we hear about the naturalist who say like our original food, indigenous food is a medicine. We should go for that. So I think for me, like creating a balance between the two kind of developments, the scientific developments, the genetic engineering, as well as the natural growth and ecology and environment friendly food. So can there be that kind of a balance? I'm sure along with the technical aspect of the today's topic, Professor Tamang will highlight how to go about this. There should be a kind of sort of coordination between the scientist and the policymaker so that a right balance is created. With these words, I welcome all the participants to share, to participate in this seminar, and hope that the kind of enlightened discussion Professor Tamang will make will surely enlighten us and lead us for the future. Thank you, and over to Pratima. Thanks.
Thank you, sir. Now, an honor for me to introduce the speaker for the day, Professor Dr. Jyoti Prakash Tamang. Professor Dr. Tamang is presently IC Mode Mountain Chair for the year 2019-2021 and is the senior most professor in microbiology serving in Sikkim Central University, India. He is a pioneering food microbiologist working on ethnic fermented foods and beverages of the Himalayas for 34 years, focusing on sequence-based taxonomy, foodomics, gastronomy of Himalayan ethnic foods, bioinformatics, food safety, nutrition, and health benefits. He has received several national and international awards, fellows of several academics, and members of national and international fora in food science and microbiology related committees. He has published more than 163 research publications, including 7 books from Taylor and Francis, USA, and Springer Nature, Singapore, with cumulative impact factors of 180, citation index of 4800, and H index of 37 in Scopus. He also has 1 patent. May I now request our honorable speaker, Dr. J.P. Tamang, to begin his presentation. Over to you, sir.
Thank you very much. I think I will just hit allow. Okay. Is it okay? Can you see?
Yes, sir, we can see.
Okay. You can see?
Yes, sir, it's visible.
Yeah. Yes, I'm audible?
Okay. Yes, you are, sir.
Am I? Okay. Now, let me first start my thanks to my old seniors, Professor Dakalji and Dr. Jaishree Pradhan for inviting me to this webinar. And I also extend my thanks to all the faculty members whom I do not know now, but I'll be visiting your department in once the lockdown is lifted. So I thanks all of them also. Then it's a great pleasure to meet. I think we have not being physically seeing each other. But I think through this webinar, my message, I would like to try not only to give you the highlights on the latest development in the field of food science or the foodomics, but also some social issues which are pertaining in the higher educations in our regions. I think within, I may take 1.5 hours, and once I finish my talk, then I request all the participants to raise the questions. I'll be happy to answer. The development in the field of the meta-taxonomic and foodomics of Indian ethnic fermented foods from past to present. So before we go, we have a global food cultures. It's divided into 3 stable food. Rice is a stable food in the eastern part of the world, and followed by the soybean, both fermented and unfermented. The fish and the alcoholic drink starts from the part of the eastern India to Southeast Asia, China, Korea, Japan, and all. Then if you go to the west, right from the Uttar Pradesh, the Western Himalayas, Western India, Pakistan, Afghanistan, then entire Europe and America, it's not the rice, it's a wheat or the barley is a stable food, bread or chapati or naan, and followed by many milk and milk products like cheese, yogurt, and the meat, hams, sausages, salami, and the wine. What is wine? Any grape is fermented, it is wine. If the apple is fermented, it is cider. So wine is not an Indian culture. So, and if any fruits are without fermentations, make it in the liquid. That is what we call the juice. It is called juice. If it is fermented, it is wine. So that is not our culture, though now wine and juice are very popular in all over the world. If you go to Latin America, the South America and the Africa, it is the sorghum or the maize is a stable food, followed by the milk and the alcoholic beverages. Now, if you go in the the gastronomy is the study of the food cultures, the food cuisines, the culinary. So rice is a stable food, as I told you, in the northeastern part, followed by the many soybean or the fermented soybean, milk product, alcoholic drink. And if you go in the West Bengal, Odisha and all, rice-based stable food is popular, followed by the fish. And in the western part of India, it is a wheat, is chapati or naan is the stable food, followed by the milk and the milk products. And in south, it is rice, but it is mixed with a legume that is called the fermented rice legume, that is dosa idli, the stable, followed by the milk and the milk products. Now, before we go to the science, let us go to the history, the past. The Indian food culture and the history. Indian foods have been well documented since 6000 BC, that means more than 8,000 years ago. In chapter 17, text Sloka 8 to 10 of Bhagavad Gita, the foods are classified into 3 types, that is Sattvika food, Rajasic food, and Tamasic food. Sattvika food denotes the food for the prosperity, longevity, intelligence. Rajasic food is the food signifies activity, patience, and tamasic food is the intoxicating. Now, in the present context, if you interpret, Sattvika food is the immunity booster, the food which boosts our immunity. These are mostly the Indian foods and all. The Rajasic foods are mostly the pickles and all, and tamasic foods are intoxicating, which are not good for the health. The Indian food culture and history. Religion influenced the Indian dietary culture towards vegetarianism due to advent of the Buddhism during 600 BC and the Jainism during 700 BC. The influence of the Ayurveda Ahar, which is mentioned in the Ayurveda, comprising mostly milk and the milk products, cereals or chapati or cooked rice, what we call chawal, and the lentils, what we call dal, fruits, vegetables, has been observed in Indian dishes, Indian diets. Now, Indian food habits, the culture has been influenced by the foreigners. The invention of the foreigners has drastically influenced the ancient food cultures too, sometimes by accepting or sharing the foods of the different communities. Sometimes we have to accept their foods into our food, or we had to share the food to them. So that is a culmination of the Indian food. The Mughal Empire, which ruled India for more than 2,000 years, has introduced the foods such as biryani, Mughal paratha, moat masala, and kebabs, and these are all now the Indian foods. The British influenced the ethnic Indian foods and supplemented many Indian foods as Anglo-Indian foods. Uh, they have ruled almost 200 years, and they introduced the bread and the loaves, which are completely unknown to the Indian food culture. Soup, salad, fried poached eggs, butter, potatoes, and so on. And in the alcohol, like whiskey, brandy, rum, and wine, were completely unknown to Indian food cultures before they came. So now it has been a part and parcel of the Indian food. Whatever it is, but the is a blend of the Ayurveda Ahar. But one thing is very important, that what it has been shown, that the ethnic fermented food has not been influenced neither by the Mughal, neither by the nor by the British. So it remains as it is, as a the ancient Indian food culture. These are the Indian thali cuisines. These are the Gorkha thali as a rice, and this is dedo. I think most of our youngsters have not seen this dedo. Dedo is a very good. It's a gluten-free now. It's a highly, the nutritious food. I think we must have. I always have the dedo every week, and I encourage all of them to have this dedo, because the concept of the dedo, say dedo khane चाहिँ गरिब को खाना हो भने, I think that is wrong. The garib dhani in the food is nothing. हामीले भन्छौँ जति पनि यो गुन्द्रुक किनेमा यो ढेडोहरू चाहिँ गरिबको खाना. But in the scientific interpretation is, what do you mean the rich food and the poor food? The the food which contains the high bioactive compounds, the immunity boosters, which promotes your health, prevents you from cancer, some other fatal diseases, lowers your sugar, then controls your blood pressures. That is a rich food. That is the food which usually it's a garibले खाने खाना ढेडोहरू जैसे भातहरू गुन्द्रुकहरू. There are fried foods, the Kentucky chicken, then hamburgers, all these things fried and all are, though it is classified as a rich food, actually these are for the nutritionist it is a poor food, because that hampers our health, that causes these so many diseases and all. Now this is a Bengali thali, as you know, they have a fish, and this is a Kashmiri thali because they prefer the bread, and this is a Manipuri thali. Manipuri thali is also very, very rich. They have many fermented and non-fermented fish and bamboo products and all, and this is South Indian thali. Now this is a European American Australian meal. As I told you that rice is not a stable food, but the bread is stable food, followed by the many hams, sauces, salami and all. And this is a Japanese meal. Japanese meal is one of the best nutritious food in the world, because these are less oil, high protein, less calories, many bioactive compounds and all. And this is Chinese food, and Korean food also very equally important like Japanese meal. Now there are three types of the feeding habits when we take the food. The first is by hand. The practice of washing hand and mouth has been mentioned in the Vedic period, and it has been mentioned in the during the Lashavi dynasty in 100 to 880 BC. So it is all documented and all. And one very important observations what we have seen is that the Indian culture typically, traditionally, we sit on the ground, cross our leg, just like this, and that is what we call in the in the yoga is the sukhasanas. So sukhasanas means when you sit here, then which is said to massage your abdominal muscles, activate blood circulation in the lower part of the body to increase the palatability. I'll explain you this. Now I'm talking. I just before the thing, I just have some snacks and all. So it has gone to my stomach, abdomen. Now there is a physiological process. all whatever I have eaten that has been processed into the liquid form, either the blood, the vitamins, or whatever, then it will be circulated to each and every part of my body and all. Now I'm not feeling anything. I'm not feeling anything. I just only feel that I had a food, I'm full. मलाई भोक लागेको थियो, मैले खाएँ, ठिकै छ, तर भित्र के हुँदैछ मलाई थाहा छैन. I do not know. So that is a beauty part of our nature. If our stomachs acts like a grinder, the grind गर्ने या त ढिकी जस्तै कुट्ने हो भने, I will have a pain, lot of pain inside. So I am not able to speak. म तपाईहरुसँग बोल्नु नै सक्ने थिइन होला. तर our stomach is such a way that they have been, it's just like a stomacher. That means the abdominal muscles play in such a way there is no pain at all. It acts, all the complex matters being grinded into the liquid form, and it sends whatever the thing. Protein is digested into the amino acid. The fatty acid is in the different type of the fatty acid, amino acid, essential amino acids, all carbohydrates, everything at the glucose form. Starch is being broken down to the glucose form, and it is being circulated to the body and all. So what is important in the Sukhasana is that will help the muscles to activate more, so it increases more digestibility. This has been proved. Another type of the feeding is the chopstick and all. This is how the Chinese have discovered an Oriental, and it has been actually discovered during the Shang Dynasty and the Han Dynasty. Initially, it was given only to the aristocrat family. The Han Dynasty has made it to the commoners also. You know what is when the civilizations, when the human life have existed in this earth, there was no fire. The ancient people had to survive by eating the raw material, the raw meat, raw fish, raw vegetables, raw fruits, whatever the thing. But when after the discovery of the fire, that they started roasting and cooking the meat and all. So when there was raw, they did not need anything to grab it because it's a raw, it's a cold, nothing else. But when it is being roasted in the fire, there is a hot. It became very hot. So they try to grab it. So they use the wood and all. So wood was not sustained for this because it got a fire and all. Then they started the bamboo, the unbifurcated bamboo and all. Then somebody said that let us bifurcate it. Then just like a thing, then this is how the chopstick has discovered 3,000 years ago. After that, the latest is the cutlery, the kata chamchi. So I just want to show that it has just discovered 1,000 years ago. So before that, there was they used to have some other means and all, maybe hand and all, but it has just in history, it has just been discovered. It has just shown that it is just an 1060. That means almost 1,000 years ago. And now, what is important is that today there are three feeding habits. One is hand by chopstick and all. If you see the hand is the oldest, more than 8,000 years. Chopstick is 3,000 years, and the cutlery is just 1,000 years. In the name of the civilization, everybody prefers to have the spoons, fork, and the knife. But they never. Most of the youngsters, they don't like to have use the hand and all. That is the choice of the every consumers. I will not say anything. I will not make any comment on that. But if you see the history and all, you see that by hand is on the oldest, and it is our culture and all. The history, the dosa has been recorded in almost 1st century CE, then idli almost 220 CE, then dokla is almost 1,000, jalebi is also more than 1,000, and papad is more than 2,500 years ago, and dahi is more than 8,000 our dahi, which is being mentioned in the Rig Veda and Upanishad, and Then the alcoholic drink, somras, it was called the somras. So it was also there in the part of the Indian culture. The drinking of the alcohol is also part of the Indian culture and all. It is mentioned in the Vedic period and all. High alcoholic distilled liquor called the Sura and the Parishad has been recorded in the 1500. That is almost 3,000 years ago.
Hello, excuse me, sir.
Now, hello.
It is not audible,
Whatever.
so please, will you please increase the volume? Will you please
No,
increase
no,
the
no.
volume?
No, I have increased it. It's okay.
Sir, can you hear me?
Okay.
Sir, can
It's
you
okay.
hear me?
Yes,
Yeah,
yes.
you please increase the volume. Everybody is saying it is not audible.
No, it is full volume.
Okay.
My
It's
volume.
near to the thing.
Is it okay?
Okay.
Is it audible?
Hello.
Hello. Is it audible?
For my
Hello.
part, it is audible. For others, it is not audible.
I think it's their connection problem, ma'am, for those people.
Oh, okay.
No, not only connection problem. It depends on the system also. What system?
Maybe. Yes, sir.
Is it okay? Audible?
It's audible, sir.
Okay. Now, our kinema, the fermented soybean food, is more than 2,000 years ago. Then historical description of the Shang of the Chi has been written in the book written by the Hooker, 1854, Risley, Gorer, and even the Risley has mentioned that the Marwa Chang is a kind of, I'm quoting it, Marwa Chang is a kind of the beer brewed by everyone in Sikkim and might be called their staple food and drink, which was recorded in 1828 in the Gazette of Sikkim. So what is fermented food? Any raw material, whether it's a plant or the animal, is subjected to the microorganisms which produce the enzymes and produce a product which has the taste, flavor, color, and it is culturally acceptable. That is called the fermented foods or the beverages. There are more than 5,000 varieties of the fermented food in the world, major and minor, and which are consumed as a staple, curry, side dish, fried, paste, seasoning, and so on. Daily per capita consumption is 5 to 40% daily intake. In India only, there are 350 major and minor ethnic fermented foods, and most of them, 50% are from the Northeast India. So there are 10 different types of the fermented food: fermented vegetables, legumes, cereals, milk, fish, meat, root, amide starters, beverage, and miscellaneous, like your chocolate also. There are the three different the characters. What I have termed is a 3A: is acidic, alkaline, and alcohol. The food is classified on the basis of the pH. The neutral pH is 7. If the pH it goes below is acidic. So like gundruk, kimchi, and all. If the pH goes up like 7.5, 8, 9, it is alkaline food. That includes the kinema, natto, and all, and alcoholic fermentations. I think I'm not going into details of this. There are two types of the identifications. What we do is that is a cultural method. Cultural method is when you get the samples, make it dilute and dilutions and all, then plating and the different media, and you see there's some colonies there. Then you pick up that colonies, purify, and you identify whether this belong to the lactic acid bacteria or the bacillus or yeast or the fungi and all, depending only those who could grow on the medium and the particular temperatures and all. That is called the culture dependent. But after then after some time, the scientists said that no, there are some bacteria. There are many organisms which are not being able to grow in the media or the cultural media. So they are present in the food or in the environment. So how to profile them is called the culture independent technique. In this technique, we isolate the DNA directly from the food sample. From kinema, if you want to study the kinema, we get 1 gram of kinema or less than 1 gram of kinema and homogenize, and we extract the DNA directly from kinema. So DNA carries all the information, all the microorganisms present in kinema, and we do the technique that is high throughput sequencing, shotgun sequencing. There are so many techniques. and all, then we can get the entire profile of that also, just like a human being. If you want to know my DNA, just get a blood of that and just do some high throughput sequencing and some other techniques and all. Then you can get the entire profile of mine, my all genetic information and all, you'll get it and all. And that will be decoded by the machine learning. That is what we call the bioinformatics and all. I'm going to that. This is the development in science. So, as I told you, the microorganisms play a very important role. They enhance the nutritional value, fortify with the health promoting benefits, produce some antioxidant, which is very important for the preventing the cancer, the antimicrobial compounds, the harboring probiotic functions and all. So these are the different type of the microorganisms. I'm not going into this. These are the lactic acid bacteria. These are the bacillus, micrococcus, then other bacteria, then the yeast. The main role of the yeast is to produce the enzymes, amyloid lactic, that is breaking down the starch to glucose, then alcohol production, sugar fermentation, the enzymatic activities, production of secondary metabolites, then inhibitory effect against the mycotoxin producing molds. Then there are mycelial fungi or filamentous fungi, and they also have the role and the production of enzymes and degradation of the anti-nutritive factor, thus improving the bioavailability of minerals. Now, in the human gastrointestinal tract, there's a gut microbiota, houses more than 10 to the power 14 million microorganisms. Our body, right from the brain to the toe, every human being has his own microorganisms. Corona is also microorganism, is a virus, one of the smallest microorganisms in the world, smallest, and the most deadliest also. The smallest organism in the world and the deadliest also. So similar type of the virus, which are not pathogenic, which are not harmful, are also present in us, in our body. So all microorganisms are not the bad guys. There are good guys also. We work on the good guys. So bacteria, some bacteria, they cause cancer. The virus cancer. Some bacteria cause the tuberculosis and food poisoning and all. But the bacteria, some bacteria are very good, which are present in our human being. That is what we call the human microbiome and all. This is what is now, there's a phyla, family and all. But still, this has to be identified and all. Just like the human being. Some persons are very good, some persons are bad. It's a human being. Criminals are also human beings. The preachers or the pandits are also human beings. Teachers are also human beings. The criminals are also human beings. Murderers are also a human being. It varies from the thing, just same like in the microorganisms also. So if you go in the gut microbiota, as I told you, these are the microorganisms, organisms which are present in our stomach, and they produce useful substances, inhibit colonizer pathogens, they integrate the epithelial layer and enhance the immune systems and all.
Hello,
yes,
sir.
Yes.
Sir,
not audible.
Can you start again? Your slide is not moving, so please start again.
No.
Can you start again?
Yeah, it's okay.
Yeah.
Oh,
yeah. Okay.
Okay. Is it okay?
Yeah. Okay. Now.
No. I'm not moving the slide.
Oh.
I'm not moving the slide. I'm just coming to I'm coming to the next slide. Is it okay?
Yeah.
Is it okay?
Okay. Yosna, is it okay? Yosna, is it okay?
Sir,
Yosna.
okay, I'm telling sir, your slide cannot be seen. Your slide cannot be seen. You have to start presenting. Presenting start गरिदिनुहोस् न.
Okay. Good. Okay.
Only your video is visible. Your slide is not visible. That's because others are presenting in the middle, so that disturbs your presentation. Okay, sir. Sorry.
Where is how to do that? Yeah, somebody resume your presentation. Resume presentations. Is it okay?
Present. Yes, sir. Now it's fine.
Okay.
Fine now. Yes,
okay.
Sir.
Is it okay?
Yes, sir. Yes, sir. Now it's
Is
okay,
it
sir.
okay? Okay, okay,
Yes, sir. It is okay now.
okay. Now this is a gut microbiome. Now the nutrigenomics. What is nutrigenomics? Is the study of the effect of the food and the food constituent in gene expression and how genetic variation affect the nutritional environment. Nowadays, the concept of the balanced food, like the protein, carbohydrate, is gone. Now we go on to the molecular level, that the biotic compounds, and which gene is responsible for which nutritional values and all. So we go in the metabolites, the proteins and all, amino acid and all. So this is the latest type of the science which we call the nutrigenomics, and the photomics is a new discipline that studies the food and the nutrition aspect through applications of the advanced omics technology. Omics is the bioinformatics, which in turn improves the health and the consumers and all. So I'll just explain you that the photomics, it includes, there are so many parameters and all. I'll tell you that one of the very important components of the photomics is the metabolomics. Metabolomics is the study of the small metabolites present in the food, that it was advanced analytical technique, data processing, and bioinformatics. There are two types of metabolomics. One is the targeted and is the untargeted. So these are the main bioactive compounds, and it has been done by the extraction of any food sampling, and by use of the spectrophotometry, that is GC, LC and all, then get the data, and we do the trivariate analysis and all, then used by the bioinformatics only, then you can interpret and all, then which genes are responsible for this untargeted metabolisms we can do. This is the latest science, and as I'll explain that, what I have explained is metabolite extractions, data acquisition, mass spectrophotometry. It may be the GC, it may be the LC, and maybe the combination of all. Then you get a peak and all. Now this peak has to be interpreted in the readable form. That is the trivariate analysis and all. There's a systems for that, and we identified into a readable form, and this is being interpreted in the KGG pathway. There is a pathway in the machine learning, and there are genes which will be sequenced by the high throughput sequence or the shotgun sequencing, and we can know that which gene is responsible for the synthesis of which amino acid, which bioactive compound. This is very important. Nowadays, this technique is also used to detect the cancers and some disease also. It's a medical also. Actually, it is being used in the food, but now the medical scientists are also. It is mostly used by the microbiologists. It's the latest development in the microbiology. We are also doing in our lab, and I'll show you some of the data which we have done that. And this is the bioinformatics, is the collection, classification, storage, and analysis of biochemical and biological information. So it includes the computer science, chemistry, biochemistry, and all. The most important component is the bioinformatics is the machine learning. So in the computer is the artificial intelligence, So whatever the sequence, whatever you do with the DNA, it is all decoded. It should be coded. It should be decoded in the readable form. That readable form can be done only by the bioinformatics, that is the machine learning. What is quite difficult to take, and it has got its own language and all. We call it the language. So that has to be fitted, and it will come into the taxonomical way and the readable form. And the bioinformatics is very important, not only in the food science, in the microbiology, it is also important in the medical science. Now, these are all the definitions of the metagenomics, high throughput. So I think I'm not going into details of this and all. Then this is the protocol for the study of the fermented foods. Anyone is interested to that, I have written an article. You can follow this and all. So this is the clear picture how we do that. Ethnomicrobiology practiced by the ancient people through the applications of the culture dependent. Taxonomy is the classification, identifications of any plant, animal, microorganism. Meta-taxonomy is by the omics, by the gene. Once you have a high throughput sequencing, and it will be given into the bioinformatics. You have it decoded and all. It is used by the omics. So I have just presented here OM. Though OM and omics has no relation at all. But if you go to the mythology, OM is very important in Hindu also, in Buddhist also, and everywhere. When you pronounce Om and all, it believes that it gives the strength, and your all muscles or your all cells will be activated and all. Then it will be in the active form. That is what we believe and all. So in science also, omics, we have a DNA, everything and all. And DNA is, we can sequence the DNA, but if it is not in the active form, it is not really decoded. So without the help of the omics, this is omics, bioinformatics, you cannot come to the real picture or the readable form. So Om and omics, as my assumption is that interrelated and all. So we get a genetic resources and you can get the whole health benefits also. These are all the steps of that. I'm not going to, this is every step of the bioinformatics, and those who study the botany or geology, they should have a knowledge in the bioinformatics. Nowadays, without the application of the bioinformatics, your papers cannot be considered for the publication of high good impact factor. So knowledge of the bioinformatics is very important. This is how we do it and all. This is then after that we do the predictive. A predictive means a hypothesis. And in the particular bacteria in the gene, which is responsible for the metabolic pathways, which is responsible for the amino acid synthesis, proteamic acid synthesis, some particular bioactive compound synthesis, that we can do with the help of the bioinformatics. That is called the predictive functionality. And on the other side, we do the real analysis of the metabolite, and we combine that. And once you combine, we'll get the real picture of any food or any organisms, that this organisms is very, very functional. That is, then we can prove that it can boost the immune systems of the human being, or it is part of health benefits and all. So now I'll go into the cultivation of soybeans, that is kinema. There are two types of varieties of soybeans, and these are the, as everyone is familiar, this is the kinema, fermented soybean food of our region, and similar product in Hawaijar in Manipur, Turangba in Meghalaya, Akuni in Nagaland, Perian in Arunachal Pradesh, Bekan in Mizoram. So these are all our publications. Now, if you see that diversity, that it might have originated in eastern part of Nepal and Bhutan also, kinema is very popular. Arunachal Pradesh, they call it Perian, Akuni, Nagaland, Hawaijar, and all. In Myanmar, in January I visited, they call it Pibo, and all. Very interesting observation is that in 8 states in Northeast, the fermented soybeans are not being prepared or consumed in Assam by Ahomi and Tripura. It's very interesting. Of course, Tripura is dominated by Bengali, but I think we are still finding that some tribes which are living in Tripura maybe, but till now we have not been documented yet. But in Assam we visited all, only they consume in the bordering with Bhutan, with Arunachal Pradesh, but not in the mainland Assam. I do not know why. The food anthropologists, I think they may have some answers. Now I come to the real science. We get a kinema, as you know that. That is how we start from the past to the present. And we use the latest the technique, that is shotgun sequencing by nanopore. Then I extract the DNA and all. And it gives the picture of the whole. The kinema, our kinema, contains the bacteria, 93%. Eukaryotes, that means the yeast and the fungi, 4.8, almost 4%. And the viruses also. Of course, not corona, but the good viruses. The good viruses. Corona is a bad guy. The good guys viruses are also present, and there are unclassified archaea and all. So this is a latest technique which can give you the entire picture of our thing. These are our resources. These are our resources. So now another technique is the high throughput sequencing and all. So this is how the kinema is prepared and all. We collected from Nepal, Bhutan and all. Then we did an entire picture of the microorganisms. Like these are the phyla, these are the genus, this is the families, these are the species level and all. The bacterial community, and all these are the different types of bacterial community, and even the fungal community also, that is the fungi and the yeast also. So I'm not going into details of that. The uniqueness is that some kinema of India, kinema of Nepal and Bhutan has a unique genera. So these organisms are very unique to Nepal. These are very unique to the kinema of Nepal. These are very unique to India, means Darjeeling and Sikkim, which are not present in the order. These are also very important bioresources and all. Then same in the fungi also. And these are all the diversity and all. Now these are all the different of the predictive functionality on. So when I was doing my research 34 years, almost 30 years ago, I could identify only Bacillus subtilis 1. But after 34 years, the science has developed in such a way that now we are, by using the shotgun sequencing and the arthropodship, now we could get more than 27 species in the same kinema. Maybe after 10 years, maybe after 15 years, we may get, maybe there will be the development of science and all, by the omics and all, we may get a different type. Now, what I want to tell you that the science has developed in such a way that it has got different paradigm shift in the food science and in our ethnic foods also. So this is a very good, the model which we have made it is that the bacteria, the microorganisms which are present here, and these are the different type of the metabolic pathways. Which bacterium is responsible for which metabolic pathways, this picture will go. So when we design for the functional food, I think this model is very important by the going into thing. So recently we have done the whole genome sequence of the organisms, and we found that the protein, this red is the indicator the immunity protein. So these are the immunity booster in kinema. So I will say that the consumption of kinema will boost the immunity, but I cannot claim that it may help to prevent the corona. That we have not done, because I'm just talking about the evidence science. But as far as our research concerned, that kinema has the immunity boosters. So it is proved. We have published a paper also. This is the immunity by whole genome sequencing. The red indicates the immunity protein, which is present in kinema. So these are all the highlights of all. Then kinema has a glutamic acid also, which is very important for the health benefits. And kinema has so many health promoting benefits. It is the source of the plant protein. It contains all amino acids, linoleic acid, and it is very good for the cholesterol lowering effect for the heart patients, vitamins and antioxidants and all. So this is the overall picture of the summary of kinema and all. Now the similar product of kinema in China, it is called Daoji, Tuanao in Thailand, Pibok in Myanmar, Chunggokchang in Korea, and Napo in Japan. So this is the production area of that. And this sticky type fermented soybean, similar to kinema, are being prepared and consumed only in this country. So I have made it kinema natto Thornau triangle. This is the hypothesis which I proposed in 2010. Is highly acceptable now. And we collected all the samples. We did a 16S RNA sequencing, and we found that there is a similarity in the Bacillus 99. That was done in the 2002. Now, at present, I told you that the science has developed. Now there is the technique of the shotgun omics and all. Now we are trying to do analyzing this. We have almost come. Because of the corona, there are some disturbances. But maybe within a year and all, we come with the all sequence, and we can say that whether kinema has diversified to NATO or NATO has come to India. We can give this the evidence science by using the shotgun sequencing and using the artificial intelligence. That is a machine learning technique. Now there are other fermented soybean products of things like soyu, tempeh, soya sauce, jumjum, which are not produced in India and cannot consume, like miso, daoji, and all. So these are the primary. And the similar type of the fermented, not soybean legume, is in Africa, because Africa they don't have they don't cultivate the soybean. Nowadays they cultivate traditionally. So they have a African locust bean, which is very similar to kinema. They have, and the taste is almost like a kinema. Now I go to the fermented cereals. that are sourdough, naan of India, dosa of India. And very interesting thing, naan is very popular in Japan. It is being, I have taken this picture and all. This is written in Japanese. They are sold in the departmental store. And naan is also popular in China. They call it nanu. I've eaten this when they served it. Then I said that it is just like our naan. Yes, it is Indian naan. They call it nanu, and naan is very popular and all. What I want to show is that our Indian foods are also being accepted by the foreigners. Just like we accept their food like kimchi, soyu and all, they also accept our food and all. So these are the other food like idli, kalam, and chira of Himachal Pradesh, gulgule of Himachal Pradesh, jalebi, and all. Our celebrity, we have already worked on this and all. Then there are many fermented vegetables like gunduk, sinki, and which are very similar to kimchi of Korea, saukai of China, sauerkraut of Germany, and this is the traditional method of thing. As I told you, the gundruk might have originated from the gunu, meaning the dried taro stalk in Newar language. So probably it is the Newari food. It might have originated from Newar. Then this is the sinki. There's a peat fermentations, as you know that, and these are all our publications and all. And there are fermented bamboo products. The word meshu is derived from the Limbu language. Probably it is a Limbu food, which mi means young bamboo shoot and su means sour. And these are the other fermented bamboo shoot in northeast like soybean in Manipur, and Yup, Hiring, Ikum of Arunachal Pradesh. And these are all our publications and all. There are fermented milk products like yogurt, cheese, kembembadi and all. And then India, we have a churpi, dahi, lassi, srikhand, and these are all the traditional method of. So this is also the technique 16S RNA sequencing. We have identified the diverse type of the lactic acid bacteria and all. So we have done the culture dependent and the culture independent also. These are all the publications and all. And the very important, the role of the bacteria present in our fermented milk product, like mostly the churpi and dahi, is that they have a probiotic genes. What are probiotics? Probiotics, when I told you that we already have a gut microbiota and all. So when there is some disturbances in our gut microbiota, we feel the gastric, we feel the constipations, we feel the stomach ache, and even it goes to the ulcers and even go for the colon cancers and all. So to activate them, to increase our immunity, we need some bacteria which have a probiotics and they go inside. All bacteria cannot survive because our stomach is very high bile content, very low pH, and all microorganisms cannot survive that. And they have to adhere it because our stomach is so slippery, our intestine. So these bacteria have the hydrophobic capacity, that ability. They can adhere to the intestine cells, epithelial cells, and they go and they activate it. This is what we call the probiotics, and which are found mostly in the milk product, mostly in the dahi and the churpi. So this is the proof, evidence-based science. We have found this, some of the gene present in that. And these are the highlights and all. And I told you that we have made the model for this also. And these are all from the whole genome sequencing, that what are the different genes responsible for the probiotics and all. And this is the sketch of that. The milk, it protects the diarrhea, allergy, reduce the serum cholesterol, prevents some of the colon cancer, elevates lactose intolerance, stimulate immune systems and all. We do not have the pure probiotic. We have a Yakult, the Japanese company, which has already come in India, in Calcutta, Delhi, in departmental stores, which are being sold. But in Darjeeling and Sikkim, it has not come. Though I am a member of the scientific advisory committee, I have already told them, I have already visited the Yakult main research institute in Tokyo twice. And this is one of the best products. If you consume that yogurt every day, you do not have any problem at all. So I suggest you, we don't have yogurt, but always drink dahi. Everyone, you take 2 spoons of dahi. Actually, you are eating the live bacteria. If you see in the microscope, you forget about the casein and all. You say that dahi khayo bhane mota huncha, yo huncha, tyo huncha. Forget about that. Main concern is that the microorganisms. If you don't prefer to drink directly, you mix with rice or chapati. Dahi sadai khanos, see after 1 month or 1.5 months, most of your digestive problems will be solved. This is the proof, evidence, best science. And beside that, there are traditionally preserved fish like the sitra, sukuti, and all, sukako macha and all. And this is in Manipur. They prepare a fermented soybean, which is most delicious for them. And in Meghalaya, they ferment it. They call it tungtak. And Tripura, in Bangladesh, they call it shital. And these are all the dry fish being sold in the Assam. And what we have done we apply the high throughput sequencing and all. We got all the microbial, the profile of that. And this is a very unique the graph, which shows that our Citra sukuti also has a specific, the unique genus, which are present in Citra. This ruminobacillus is not present in others. So this is very, very unique and all. This is how I come to know that. If you go for the cultural explanation of how our Gorkha communities have preserved this and consumed this. I am not an inventor of this. My team are not inventor of this. We are just as researchers. The inventors are our forefathers. But they did not have any scientific basis. They did not know what are the bioinformatics, what are the shotgun sequencing and all. But by applying this the same food which we have been consuming for more than 3,000 years, and now today we can show that these food are the very unique and which goes for the immunity boosters, so many health promoting, and the bioresources, the functionality and all. This is how we have done. I just want to show that how we do the past to the present, a simple food which we are being sold in the market and come into the scientific way, and that we have proved it that also. These are all the things. Now same in the meat product also, like the American sausage and all. We also have sauces like beef sausage, the cargium in. These are all the different types of meat products and all. So we also apply the same proteomics and the the metagenomics and all, and found it. And these are the graphic how we do a science is a typical meat products, and how we do the metataxonomics using the omics and the identified, and they have a predictive functionality. What are the health promoting benefits, and which bacteria are responsible, which bacterium is responsible for which metabolic pathways we do, and this is very, very important data for the optimization for the productions of the functional food. So these are all our application. Now I'll come to the amylolytic starters, the starters to prepare the alcoholic beverages. We call it matcha. The similar product in China is Yayum, and Nurut in Korea, and Main in Vietnam, Ragi in Indonesia, and Lopang in Thailand. See, these are the starter cultures in Northeast Asia. There are hundreds different types of the starters like our matcha. In every 8 states of Northeast, they have this type of the starter cultures. And we collected all, and these are the preparation of the matcha. And in matcha, the Saccharomyces, these are the yeast, Saccharomyces, the mucor, and the filamentous molds, then bacteria are present and all. These are the filamental molds. They are all good guys. They are not bad guys. They don't cause any food poisoning. They give the alcohol, they break the starch to the glucose and help in the production of the alcohol. They are all good guys present in our starters, in our tank. And we publish the papers. These are all the publications and all. Then see, with the help of the ITS PCR, we could identify only 6 species. Then we use the DGG PCR, better technique, only 18 yeast. But by the high throughput sequencing, we could get it 83 yeast and 511 volts. So high throughput sequencing is very, very powerful tool to profile the entire microorganisms and all. So these are the the fungi, the different type of the fungi. Then these are the different type of the bacteria also. Now, this is how kodo is not so popular, but if you boil it and then mix with the starters and all, there is a saccharification. The starch is broken out of glucose within 2 days. If you taste it, it's very sweet because the organisms, microorganisms, which I showed you, they play very important. They produce the enzymes and they break down the starch to the glucose. Then after the fermentation, there is a glycolysis cycle that the glucose converts into the alcohol and the carbon dioxide, and this is how we consume. The alcohol content is only 4 less than beer, and it has contains the vitamins. It has a high calorie food. It is mostly given to the postnatal women and the ailing persons and all. And this is a traditional drink. And similar type of the product is sake of Japan, is a national drink, and the makuli is very similar to our bhatija. So our bhatija is still at home. It's not being popularized. It is not being commercialized. It is not being the thing. It is just being consumed at home, also in the rural areas, not in town. But the same type of the bhatija, they distilled and they call it the makuli. I have tested both bhatija. I have tested makuli same. But the very interesting thing is that makuli is exporting and they earned more than $10 million in 2016. Probably this might have gone now, maybe 15 to 20 million dollars